So I asked you to pre-solve, but some people don't know what that means. They think they can solve in sessions. That means if they get tired, they can take a break and then continue again. Are you those people? Did you even pre-solve? No. What? I gave you a task, bro. Okay. Did I get a 5 or a 1? It's your choice. Oh, by the way, there's something called the blueprint, okay, which is, I had this revelation the other day that everyone can get a 5 regardless of the brain or the genetics, as long as you got the discipline, honestly. So all I ask you to do is to count how many days you have from now till the exam day. And let's just be realistic, right? Don't count Thursdays and don't count, let's say, 2 days of aid, right? Let's say 2 days you get depressed. Don't count those either. 3 days of aid. 3 days of aid. So that's going to be 7 days gone, including Thursdays. How many days from now till the exam day? March is how many days? Let's just say 24, if you don't count Thursdays. Let's just say. Okay, and let's say a depressed day here and there. 24. April is how many days? 24. That's 48. Take out 3 days of aid. That's 45. Exam, I think, is on May 5th. That's 4 days. Don't count the 5th, obviously. So let's say 50 days, right? Let's say 50 days. If you are not here, don't vote. Who is this voting? Hello? Yeah. Ah. Okay. Your father. Okay, so 2 hours wins. Okay. We can always vote, but the ones who ask for the reduced time are not even here. You know, the BISC students, they're not here. Okay. So going back to this, the blueprint thingy. So the blueprint is your step to a 5. Okay. Regardless of your brain power, regardless of your enthusiasm for the subject, regardless of whether you are a grade 9 or a grade 12, just do these files. Just okay, only 2 pictures, right? So here I give you per year what you are given in the drive, right? Because some people start to look at this list in the last 10 days before the exam. They're like, oh yeah, let's start practicing, which is actually very late, obviously. And of course, some will just panic and give up. Don't be that person. So the best way not to panic is to actually start today. Okay, this is your full list. I think it shows better on your laptop, and also this list here. So now, because some people don't know what to do in the drive has many, many things, but I want you to put your focus on these things because the progress checks and the topic questions came out in 2024. They were not there previously, and their style of the questions is very different, much harder than the previous. So please. Now, I'll also actually make you College Board? College Board 2024. Like College Board, each unit has 2024, and it's going to be there. Okay. Good. Yeah. So yeah. Today we do the 2020 part 1, if we get to finish, right? And that counts as one thing off your checklist. Okay. So don't waste the weekends, by the way. The weekend is like your best time to achieve all of this. And of course, some of you will just say this is impossible, but we're not asking you for 100% on your exam to get a 5. We're asking you for a 70-something% to get a 5. If you guys go home and say to your parents, I got 17 exams, they're going to say this is horrible. And they it's a 5, which is amazing. Okay. Some will say, how can I practice if I haven't finished the units yet? Don't be that person. Don't be that person. Okay. Okay. Very good. Hope you're listening. So 2020, test 1. Let's go. Okay, so I already solved the MCQ video-wise, if you guys want to go over it again. But We'll just pause this one here. I think it's better. All right. Okay. Stop. Let's do it. Okay. Humans produce sweat as a cooling mechanism to maintain a stable internal temperature. Which of the following best explains how the properties of the water... Are they guys on Zoom? Okay, this guy doesn't join unless we say we're online for some reason. Can you just say we're online? I don't know why. No, different guy. He never joins until we say we're online. Until. Okay. So, which of the following best explains how the properties of the water contribute to the cooling process, right? So, what do you guys think? How do you cool by sweating? The body's heat is used to evaporate the water. The body's temperature drops as a result. Right? Vaporization. Vaporization. Right? If you want to cool down, do you want to absorb excess amounts of heat? Of course not. And melting doesn't have anything to do with it. Yes, you are correct. Here we have two chambers that are separated by a semi-permeable membrane, which means it only lets some things go through left to right or right to left. Membrane permits the movement of water but not salt. And what you want is to vary the amount of the water movement rate. You want to end up in a faster net rate of water movement into chamber A. To have more water go into chamber A, you also have to have more salt in chamber A, because water always goes where the salt is. So place the salt in chamber A. Okay. Was it easy for you? Is this new for you? Okay. Which of the following best describes the role of water in photosynthesis? Photosynthesis has how many stages? Two. First and? Second. Exactly. The first stage requires the water to be split by the sunlight to become hydrogen ions, to become electrons, and to become also oxygen gas. So electrons are produced by the water. Okay? So it's B. So it's B. Some would say it's A because they see protons. Would they be correct? Whenever they say only, don't choose that. Only or every or all, it's never the answer. Never the answer. Okay? Very good. Part 4 or question 4. The word compartmentalization. Who knows what that means? Membranes. Having internal membranes. Because having internal membranes is a property of only eukaryotes. One of those membranes is going to be the one on the nucleus, which separates the nucleus from the cytoplasm. What's the process called that can be used to use DNA to make RNA? Transcription. It happens where? In the nucleus. What process makes RNA to protein? Translation. So you must physically separate them. Thanks to what? Thanks to the nucleus. Okay, we have an enzyme called pyruvate dehydrogenase, which converts pyruvate to acetyl-CoA. So over time, acetyl-CoA is going to increase, and pyruvate will decrease. Acetyl-CoA is then getting broken down in the Krebs cycle. You guys must know this from background information. And a researcher measured the accumulation of acetyl-CoA, okay, under several different conditions. Good. Which of the following best describes the cellular location where pyruvate dehydrogenase? Guys, they just said the Krebs cycle. Where's the Krebs cycle at?
Mitochondria.
No, sorry. No. I made a mistake. Pyruvate dehydrogenase does its work before the Krebs cycle. Okay? Before the Krebs cycle. So, but it's still happening in the mitochondria. In the mitochondrial matrix. That's where pyruvate becomes acetyl-CoA. Okay. See if this guy's online. I need text, bro. Text. Okay. Number 6, the maximum production rate. Do you guys know what rate means? Slope, exactly. Rate means the slope. Of acetyl-CoA production under condition 1. So what do we do? How do you find the slope of that line?
65 for 90 meters. Because, you know.
Don't look at the end. Look at where it stops changing. So look, from here to here. Because afterwards it becomes horizontal.
65.
So 65. 65. Here's going to be 0, 0. 65 minus 0 over 65 minus 0, which is 1. So it's going to become 1 micromole per second. Seven. Which of the following observations provides the best evidence that acetyl-CoA negatively regulates pyruvate dehydrogenase activity? What does negatively regulate mean? If there is more of something, there is less of something else. That means if the acetyl-CoA increases, the pyruvate dehydrogenase activity decreases. Okay. Do we see that somewhere here? A, right? The rate of the catalyzed reaction by pyruvate dehydrogenase becomes less when there is more acetyl-CoA. Pyruvate dehydrogenase deficiency is a genetic disease most commonly linked to a mutation in the alpha subunit of the mitochondrial enzyme. So if you have a mutation in a mitochondrial enzyme, you will end up with pyruvate dehydrogenase deficiency. As a result of the mutation, affected individuals build up dangerous amounts of lactic acid. Of course they do, because if you don't have pyruvate dehydrogenase, you don't have a way of making pyruvate into acetyl-CoA. So you're going to have a lot of pyruvate, and the body needs to get its ATP. The pyruvate then has to get used up in fermentation instead. So now the body has to go through fermentation, lactic acid fermentation. So what do we think is the answer here?
D.
The diagram shows how water can adhere to the xylem in the stems of plants, which contributes to water movement in the plant. Which of the following best explains how water is able to move upward? Guys, for the water to actually form Do you guys know what this is?
Hydrogen bonding.
Exactly, it's a hydrogen bond. And for the water to actually move upward, it must hydrogen bond with two things, itself and also with the xylem. So to hydrogen bond, you have to be polar. So it's water is non-polar. Sure about that? I heard it be from someone. D as in dog?
Yeah.
Okay. Protein X activates gene expression only in cells exposed to a specific signaling molecule. Guys, if protein X affects signaling molecules, what do we call protein? Justin, as a host, you must, before, I mean, it's going to become super cold. Guys, let me help you. Here you go. I mean, at least say thank you.
That's not
It's not Ramadan yet.
يعرفني ما أحب الشاي.
Okay. So, if protein X is responsible for the activation of signaling molecules, what do we call protein X? Like, what does it has a name? It's made of two words, that name. Exactly, transcription factor. That's correct. In a study, researchers determined the intracellular location of protein X in cultured cells, both before and after exposing the cells to the signaling molecule. Okay, so before the treatment, protein X was found in the cytoplasm, but after the treatment, it was localized where? In the nucleus. What's the answer? Exactly correct. Very good. The hormone prolactin has varying effects in many different animal species. We are vertebrates. We have prolactin for making milk. In fish, it is used for regulation of, it's called osmoregulation, which means regulating the concentration of your blood as an animal, as a fish. In a bird, it's used for breaking down lipids. So in every vertebrate, it has a different function. Okay. So guys, all vertebrates have it, yet it has different functions. That means the same common ancestor, but different functions. Do you guys know what that's called? Homologous. Homologous structure. Okay? Exactly correct. Same common ancestor, different function is called homologous structure. Easy as that. That means different common ancestor, but similar function. So it's the opposite.
And different structure.
Different structure. Okay, this actually is a hard question. Certain reef-building corals contain an algae that can make DMSP. Now, DMSP is important. It goes into the water, then becomes DMS gas. So DMSP becomes DMS gas by microorganisms. Then it's going to enter the atmosphere. It's going to trigger the formation of sulfate aerosol, which means cloud formation, which will block sunlight, heating the water. Okay. This symbiont algae produced DMSP. When there is high water temperature. So the high water temperature makes the algae produce DMSP. If the water temperature is too high, the corals will have to expel the algae that make the DMSP. Now, researchers measured the amount of DMSP produced by juvenile, which means young, and adult coral and their symbionts. What's a symbiont? It lives and interacts with you. Could be helpful, could be bad, could be, many different things. But I'm assuming that algae is a good thing because they can, you know, probably do photosynthesis and give you food. Okay. They do it under two conditions. Normal conditions, which is at 27 Celsius, and also thermally stressed conditions, which is going to be 32 Celsius. Data are shown in the graph in figure 1. Okay. Let's just read the graph for a bit. In figure 1, okay, it's both figure 1. Left side, we see that as days go by, the 32 Celsius is moving up, and it's going to have more and more DMSP concentration compared to the 27 Celsius. And the same result goes for the adults. So basically, DMSP is higher under 32 Celsius for both juveniles and also adults. Now, the researchers measure the density of the symbiont, which is the, I think, algae, probably the algae, and also the photosynthetic yield in adult corals at 2 different temperatures. Now, what is this yield? It's an index measure of energy output. Okay. So what do we see happening? When it's 0 days, there is overlap. They're not different. But at 5 days and also at 10 days, there is no overlap, which means they're different. And when it comes to the photosynthetic yield, it's going to be always different, with 27 Celsius having more yield. Why do you guys think that the 27 Celsius have more yield?
Because it's the ideal temperature that they're used to living at.
Did they say that somewhere? Because the algae is still there, still on the coral. But here, the algae is expelled from the coral. Okay. No. Question 12. Which of the following best describes the production of DMSP by coral and coral symbionts?
It's negative.
Why? How do you know so?
Because the mechanisms that the MSP produces, it prevents it.
So look, guys, when the water has a high temperature, DMSP is made in order to block the sunlight heating the water. So yes, it's a negative feedback because the high water results ultimately in low water temperature. That's called negative feedback. So it's going to be either A or B. It's B?
Yes.
Yes, because from high water temperature to lower. So it's going to be reversing it. Which of the following best describes the effect of temperature on coral's ability to produce DMSP as shown in figure 1? It seems that the higher temperature have more DMSP in both juvenile and adults, right?
So it's A.
So it's A. Wait.
Yes,
Yeah. Less at 27, which means more at 32. Which of the following best describes the difference between the total amount of DMSP produced by adults compared to juvenile at the start of the 32 trial?
adults produce 5 times more than juveniles.
How much is the juvenile?
Like 3.
Like, yeah, 3.1. Adults?
So about 5 times.
Yeah, let's just say 14. Divide by that, about 4 point, I think 7 or 6, which is like 5 times more. Okay, so the adults produce it 5 times more, right? 15. In addition to the effect of temperature on DMSP produced by corals and their symbionts, which of the following relationships is also being considered in this experiment?
Effects of additional DMSP produced by symbiont in the coral's age.
The coral's age, yes, because we're comparing juvenile to adult. That is correct. Are we varying the light intensity?
No.
No, they never mentioned light, I think, in the experiment, right? So it's not C. Are we
And A, it's not.
are we comparing the coral species or just the H?
Yeah, there.
Just the H. Which is B. 16. Which of the following best describes the scientist's finding concerning the density of symbionts presented in figure 2? Well, they are not different at 0 days. They become more different at 5 and 10 days, with the symbiont density being higher at the 27 Celsius. So algae is the symbiont. Okay.
Oh, it's C.
Yeah. At day 5 and day 10, they're different, but not at day 0. Which of the following best explains the result of adult corals being exposed to elevated temperatures for extended periods of time? Elevated temperatures. Well, they expel the algae, right?
So they obtain less energy.
They obtain less food from them, exactly. So,
C.
so it's not A, obviously. It is what?
C.
C. Okay. If you were given the frequency of the capital A, how do you find the frequency of the small a?
1 minus 0.55.
Now let's find the frequency of capital A, small a. Now, the common mistake here is that people just multiply 0.55 by 0.45.
Multiply by 2.
By 2 as well. Because capital A, small a genotype means 2pq. So 2 times P times Q. And I think you get this one. Okay. 19. A particular genetic disorder results from a single change in the amino acid sequence coded for in a gene. Let's just compare the normal DNA sequence to the mutated version. How are they different in where? In the last 3 letters, right? We had a T becoming A. Now, this is DNA or RNA given right now?
DNA.
How do you know?
No, you
Yeah.
just sign.
Very good, very good. Let's now find the RNA. So,
GAG.
GAG. And here, GUG. What's the amino acid by GAG?
Glue.
GAG, yani, basically, yes, this one. Okay. So glue. So it was glue. How about GUG? So G, U, and G, val. So it became val. So from glue to val, yep. Is this, is it recessive?
Yes.
How do you know?
Mother gene.
The mother gene? Can you speak more about that?
So it's mitochondrial,
Oh, how did you know?
because every disease affected from the mother is mitochondrial. Mitochondrial comes from the mother.
Okay. So here we have a single affected parent who happens to be a female. And guess what? All the kids are sick without any exception. And the same pattern is repeated here. A single parent who is sick, the mother, the female, all the kids are sick. But guess what? When the father is sick, none of the kids are sick. When the mother is sick, every kid is sick. When the father is sick, no one's sick. That means it's mitochondrial or chloroplast if it was a plant. It's a mitochondrial one. Twenty-one. Oncogenes are genes that can cause tumor formation as a result of a particular mutation. Okay. They can cause tumor formation. Okay. Which of the following potential therapies would be the most most effective at preventing the expression of the oncogene? Okay, so you want to prevent the expression. The best way is to go straight to the promoter. That's the best way. Go straight where to the promoter. See? I see a promoter here. Okay, that's the best way. UV. Radiation can damage DNA by breaking weak bonds. Guys, if they say weak bonds, are they discussing hydrogen bonds or covalent bonds?
Hydrogen.
Hydrogen.
Really? Which one's hydrogen bond?
Exactly. So this one or this one. The hydrogen bonds are between the sugar phosphate? No, they're between the two strands, between the nitrogenous bases. So yeah. Okay. Scientists are interested in determining the evolution of 7 lizard species found on different islands. They isolate DNA from individuals of each species and sequence the mitochondrial gene that encodes cytochrome B. The numbers of genetic differences between the species are shown in the table below. Okay, so right now we're looking at differences, not similarities. So the less the number, the closer they are to each other. So it seems like F and G are very close. The only two differences apart.
Wait, how do you like read this, John?
For example, A and B are different by 34 amino acids. A and C are different by 39, and so on. So now it seems like G and F are only different by 2 amino acids. But I don't see that. Let's continue. So C and B. Where is C and where is B? They are different by 23. Let's save the number. E and D. E and D.
Wow.
They are different by 4. That's probably the smallest. F and B. F and B are 22. G and A, 47. That's very big. So the lesser the number, the closer they are to each other. Okay. So here we have the endocrine system. Hormones are being made by specific areas like the hypothalamus or the pituitary gland or the ovary and so on. Now we're talking about a positive feedback. That means that the end product makes more of itself. Okay, so can you make more uterus? Probably not, right? Can you make more estrogen and progesterone? Probably yes, right? So these guys have to go back and make more of themselves. So do you want to inhibit or release?
Release.
Do you want to thicken the uterine lining? It's not. Okay. There's this thing called DCPIP, which is a chemical dye, which starts as blue and then is going to become clear upon getting reduced. Reduced means gaining electrons. So DCPIP will be acting as an electron acceptor. Okay. A student performed an experiment to study the effects of a chemical DCMU on photosynthesis. Okay. They added DCPIP to 3 of the tubes, and they added DCMU to 1 of the tubes only. And tube 3 was wrapped in tin foil. What's the point of wrapping it in foil?
Insulate. Isolate.
Block sunlight. There's no light here. How are tube 1 and 2 different? By DC pip. It's actually a huge difference because if you guys look at tube 1, which is the straight line, there is nothing happening, right? But look at tube 2, it decreased. So in tube 2, blue to clear. Okay.
2 or 3.
2. Okay, see. Okay. And okay. So reaction only happened in tube 2. Only. In which of the following tubes is the greatest reduction? That's obviously tube 2. Reduction means gaining electrons. And if you were to gain the electrons, you're going to become clear. And if you're clear, you're going to have less light absorbance. Absorbance only works for colors. So it's becoming less blue over time, and therefore a reaction has occurred. Which of the following claims is best supported by the experimental results?
Light is required for electron transport chain to transfer electrons.
He's right, because tube 2 does have light and does have DC pip. He's right. But isn't DC pip acting as a source of electrons? In choice D? No. Was DC pip electron acceptor, not electron source? It's the exact opposite. But some students chose that because they saw electrons, they got confused. Don't be that person, okay?
I did.
You actually did? Shoot. Okay. Which of the following best explains how DCMU affected the reaction? DCMU acted as a second buffer? I mean, probably not. DCMU, if you were to put it, you get no reaction. So DCMU blocked the reaction, which means blocked the transfer of electrons.
Are we still in 27 or 28? 27. I moved to 20.
Smart man. So blocked electrons, right? Guys, smart or what? Let's go.
Indeed.
Let's see the question, man.
I saw it. I was reading it. I went to the side. I didn't go down.
Which of the following best justifies the use of tube 2 as a control? Is it a control? Okay. If tube 2 is a control, it's a control for what? For which tube?
Determining the effects of DCMU. So for tube 3 and 4.
Look, tube 2 can actually act as a control for 2 tubes, for 3 and for 4. If it's a control for 3, it's a control for the effect of light, right? But if it's a control for tube 4, it's a control for the effect of DCMU. Look for something like that. Is about the accuracy? It's like weird. Chlorophyll suspension? The heck is that? I don't know. What's that? What's chlorophyll suspension? Exactly. Don't choose it. If you don't understand it, don't choose it, okay? Don't say, oh, it's the longest answer, has to be the right answer. That's like a rumor or something. C, it's a positive control for the change of DC pip
Color.
as color. It is changing in color.
Well, it's not a control for color.
It's not, right? It's a control for light. We're actually, oh, light intensity, not light. Like here, what's the difference? So the question says either there is light or there's no light.
Yeah.
This one says light intensity, which means that we were, like, if you choose C, that means you're telling me they varied light intensity. They didn't. They either had light or they had no light. Okay? So it's not that. It's not that, okay? And yeah. Good. So it's about the effect of DCMU. All right. Scientists designed an experiment to test an artificial membrane that mimics the phospholipid bilayer of a cell. They built a tube that was divided by the artificial membrane, and they filled it with distilled water. They put some amount of protein to the water on one side, and at some point they measured the concentration of the protein on either side. But guess what? The protein did not move. That means the protein was too large to move through the membrane.
So use something smaller, but maybe.
Yes, use a substance that is of less size than the protein, so it can actually move. 30. Which of the following best illustrates the flow of information required for the synthesis of proteins encoded in the genome of a retrovirus? What's a retrovirus? RNA virus, right? RNA virus. RNA virus has to become DNA first, and then DNA makes RNA. RNA makes protein. So choice C. Which of the following best describes the process by which gas?
So retrovirus is always an RNA virus.
Yep. Gas from the atmosphere is obtained by the plants and used to build lipids. Sulfur cycle? Never heard of that in my life. Nitrogen cycle? I mean, that does exist.
So it's not the answer?
That does exist, right? But you are building lipids. Lipids don't have nitrogen.
Carbon.
So carbon cycle. Lipids have lots of carbon. 32. Which of the following best explains how the phospholipid bilayer of a transport vesicle contributes to cellular functions? Bilayer. Okay, transport vesicle. Look, a transport vesicle has to fuse. Has to what? Fuse. Okay. Let's look for that, for the fusion part. It's not that. It's not that. It's A, right? It has to fuse. The diploid number of chromosomes in a cell is 78. So right now 2n is equal to 78. Okay. Does that number get halved during interphase? No.
Doesn't it get doubled?
The chromosome number does not get doubled.
The DNA.
The DNA gets doubled, not the chromosome number. But after fertilization it becomes halved. So look for 39. No, sorry. After meiosis, I mean. Look for 39 after meiosis. So it's going to be either B or C. How come it's not C? Sorry, yeah. Look, the chromatids get doubled, not the chromosomes. So be careful. The chromosome is like just the centromere. That does not change. Scientists were interested in testing the effects of rotenone, a broad-spectrum pesticide, which means, it's a medicine that kills pests like insects. Cell culture A was used as a control. Culture B was used to be treated with rotenone. After a period of time, the scientists measured the concentration of several metabolites in the mitochondria of cells in both cultures. What is very different between them? Which chemical? NADH.
Yeah.
It's 10 times higher in culture B, about 10 times higher. Actually, it is 10 times higher. Why is it 10 times higher? Because the enzyme that was, taking the electrons out of the NADH is not working in culture B. That's why. Does that happen in the Krebs cycle?
No.
No. So look for NADH. So either going to be. Mhm.
C.
How about B?
Transfer proteins are not blocked from entering the mitochondria.
Transport proteins are actually on the mitochondrial membrane. They don't enter. They're already there. Exactly correct.
Let's kind of repeat it.
Look at the row.
For
Hmm?
B.
For B. Look at B. They're saying that transport proteins are blocked from entering. Transport proteins don't move. They're fixed on a place. So this is wrong. Which of the following best explains why triploid bananas don't produce seeds? Because look, they have a haploid, they have an odd number of ploidy. Odd number is not going to work, right? Like if you have a 2n or a 4n or a 6n, for example, works, but 3n, like how do you have that? 0.5? Doesn't work, right? So if you make that into 2 halves, that just does not exist. So, yeah, unequal pairing.
C.
PCR is a machine or a technique that can copy DNA in large amounts. And they have to rely on an enzyme of DNA polymerase that was isolated from a special bacteria called Thermus aquaticus, which lives in hydrothermal vents. Okay. Which of the following best explains why the polymerase from the species T. aquaticus is used for PCR? Guys, this bacteria has functioning enzymes at very high temperatures because this is a very high temperature. Okay. So now what happens is that part of the PCR procedure is to actually denature the DNA at 94 to 96 Celsius, which means break it down. And the enzyme has to be able to work at this very high temperature. B. It must not denature at high temperatures.
So isn't it denaturing? Isn't it denaturing at?
Oh, they fooled you. They fooled you. It's a common mistake. They don't mean here denaturation of a protein. They mean denaturation of DNA. In PCR, you break the DNA strands by heating them. Okay, so they fooled us with that. And so, look, a machine of PCR requires the temperature to be as high as 96 Celsius. That means the enzymes in the PCR machine have to be able to not lose their shape at this high temperature. They should not denature at this high temperature, which is B. Polymerase is the enzyme. Of structural similarities as a result of convergent evolution. Guys, if we have convergent evolution, should they have the same common ancestor or a different one?
Different one.
Different one. So it's not C and it's not D. They must have a different common ancestor. That means they must have low genetic similarity. The function should be same.
Structure different.
Based on the cladogram, which of the following are the traits shared between the ferns and the pines?
Anything with wood, seeds, and flowers is out. So xylem and embryo.
He's right. Ferns have embryo and xylem, while pines have embryo, xylem, wood, and seed. They only have embryo and xylem in common. Okay. All right. Snapdragons are a type of flowering plant. The flower color of plants is determined by a single gene with two alleles. Incompletely dominant R allele and a recessive small r allele. Okay. They picked 100 individuals and they recorded their flower color. And after 7 generations, they did some work. Okay. Okay. Great. So what do we see here? Number of the red snapdragons went up. The pink ones went down. The white ones went up. Correct? Okay. Guys, do you know how to read the chi-square thingy?
Yes.
We have a yes.
No.
Yes.
No.
How many no's? That's okay.
Mr. For the 2 years that I've done AP Bio, I've always skipped chi-square.
What was the thought process behind that?
The most that can happen is one question, so it's not worth it.
It's 2 in this exam, I think.
Okay, so guys, look. Do you know how many alleles we are dealing with here?
3.
We are dealing with 3 alleles.
-1.
Wait, how are they 3 alleles? Huh? Sorry? Who spoke? Someone said 3 alleles.
I said 3 others.
Where are the 3? Tell me.
Pink and
I'm somewhere.
No, I want the alleles.
I mean, you mean phenotypes, not all these.
Guys, let me give you a nice cheat. If you see Hardy, what is this? I don't know what happened. One second. What is this? If you guys see Hardy-Weinberg, always choose your degrees of the freedom to be equal to 1. Like without any exception. Without.
Voila.
Wallah. You see?
It's nice.
Yeah. Okay, now. So look at these degrees of the freedom and just choose 1. And they want the significance level of 0.05. This is called the critical value.
Oh.
It's not wrong.
A lot is.
The answer is not A.
It's
What
B.
is it? Sure.
دقيقة 39. It's B still.
I think you're looking at different.
No, لا مش نفس الشيء.
Let's say it can't be any other.
Wait,
خليني أشوف.
are we in the same?
Mr. I have one too here.
Wait, I'm actually like scared now.
It has to be wrong.
39. Sorry, man. I'm mistaken.
إيه ليش كوجول يقول لك شو يسمونه أنت غلط؟
It's okay.
روح على كوجول لا ينشأ عليه.
No, go to the detailed answer key, bro.
إيه ليش شنو الأنسرز غلط؟
Yes.
يروح ايش يسمونه على كوجول مو عليه.
Okay. So guys, do you get the cheat? Hardy-Weinberg means that. Don't even think. Don't bother thinking.
What about if it's other degrees? How do you know?
That's a different question. You're asking right now.
Can you?
For 2 years, you start to skip it. I mean, you're skipping it. Now you decide to not skip it.
Yes.
Let's find. Okay. That's a good one. Let's finish the MCQ. I'm gonna show you a file specific for that. What do you ask for?
I've seen it, but have I opened it?
Okay. So sea otters are native to the western coast of North America. And between 1750 and 1850, that's 100 years, hunting reduced their population from hundreds of thousands to only 1,000. Do you guys know what this is called?
Under effect.
No. Other one.
Bottleneck.
The bottleneck effect. Very good. Now, in the early 1900s, a small population of sea otters was discovered in an estuary in Central California. Okay. Of course, they were now protected by a treaty, so there was no more hunting for them. But now they are only 3,000. Not so much. They already have little genetic diversity. Otters live in kelp forests, which is this guy. Where is he? Not here. Okay, the kelp is like their the producer. Okay, it's like a grass, but in the water. And then eelgrass beds feed on the 1 second. Feed on crabs on. Okay, so the otters feed on the crabs. They also feed on the shellfish, which is not shown in the figure. Most herbivores, which means plant-eating animals, in the habitat eat algae that grows on the eelgrass, like a sea slug eats the algae. Okay. If there is too much algae, the eelgrass will get harmed because they will cover the surface and cannot receive any sunlight. With more otters, the eelgrass has increased. Do you guys know what that means? That's actually like a very important sentence right there. This is like a question answered already.
Doesn't mean the otter is a keystone species?
Smart. Smart. Otter is a keystone species. If the presence of a species makes the producers more, it's called the keystone species, which answered the question. Now, recently, scientists have noticed that the presence of two non-native predatory invertebrate species, are now colonizing this estuary region. 3 decades ago, it was too cold for them to actually be there. But now, because of climate change, they have done what? They're now moving there. Correct? Now, we also have an additional problem that now the otters are experiencing increased mortality, which means death, because of more harmful algal bloom. How does an algal bloom happen? You guys know what that is?
When there's
More toxins?
No. Too many fertilizers go into the water.
is it bad? Is it a bad?
It's bad.
Mm.
It's very bad. Yes. They're called harmful.
It's caused by humans, right?
Everything is caused by humans, exactly. Okay, we are the ones doing the pollution. They like the pollution, right? And now who eats them? The shellfish. And the otters eat that shellfish. Which of the following best describes what happened to the otter population between 1750 and 1850?
We already talked about a bottleneck effect.
Exactly correct.
C.
Well,
Oh, sorry. Yeah, either C or A.
well, you mentioned bottleneck.
Wait, did they lose habitat?
No.
Was that mentioned? No.
No.
Is it a founder effect? No. Okay. They decreased in size because of killing impact, because of hunting, which means bottleneck. Based on the information provided in the passage, which of the following best describes the effect of harmful algal bloom on the otter population? The algal bloom, whether they are like, do they care about the otters, whether they are low or high, or they just do damage?
You just do damage.
Just like a natural disaster. It happens whether there is a lot of that thing or not so many of it, because the bloom is somewhere else completely. It depends on the nutrient pollution, which can happen regardless of how big the otter population is. So if it's going to be independent of the size, that's called what? Density independent factor. Okay.
How about the density dependent?
That depends on the size, like disease. Disease is one, or predation, or competition. So it's going to be density independent. Yep. As otters were removed during the hunting years, there was a large decrease in the catches of fish species from the eelgrass habitat. Large decrease in the catches of the fish. Okay, that's a bad thing. Why would this decrease happen? She already answered.
Yeah.
Otters are keystone species. Is it B or A?
B.
Why not A?
Affected the population size of another species. What does it have to do with another species?
Of one other. Keystone affects everything, the entire ecosystem. Climate change could affect the ecosystem of this estuary region in many ways. From the information provided, which of the following predictions about the direct local effects of climate change is most likely? What do you guys think? If it's becoming warmer, they can now live in that area, those invasive species, I mean, okay, which are called non-native species. So I think it's going to be choice A. I think, yes. If you look here, they said that there was the presence of 2 non-native predatory invertebrate species in the estuary in the time where it was too cold for them 3 decades ago. But now they have moved there. So if it's too cold, they cannot live there. Because of climate change, now it's warm, they can now live there. Okay.
Yes.
Based on the information, an increase in the sea slug population would most likely be directly related to which of the following? Increase in the sea slug. Okay. If you have more sea slug, you're going to have more crab and more this. You're also going to get less algae, right? Right?
Yep.
Okay.
Those C.
Yeah, because they eat the algae.
C or D?
C.
Yeah, because if you have more sea slug, you must have more crab. Because crab eats it, not less crab.
No.
Many fish species, such as fathead minnows, release a pheromone when their skin cells are damaged. Researchers placed pike, a predator of the fathead minnows, in a choice chamber, and they released the minnow pheromone at one end of the chamber. So guys, if there is a minnow pheromone, the predator thinks that there is a fish in the water, and they will go toward it to eat that fish. Okay? Which of the following questions will best guide a follow-up investigation about the role of pheromone in locating the prey? The role of pheromone. Is it B? No, I mean, they want to prey because of food, probably, right? Is it the pike having a predator? Makes no sense.
How do I determine that the fathead minnow pheromone is present in the world?
Mm-hmm. Exactly correct. Exactly.
So
Mm-hmm.
حتى تشات جي بي تي قال بي.
Tell it it's A is going to explain to you, I swear.
بس مستحيل اي.
والله.
بس انت علمتني على هاي الطريقة السنة راح يعني من وراك ماخذ.
No, Hardy-Weinberg actually has an exception. It's actually in my cheats, bro. Check out my cheats. I think it's What? One second, guys. Thing is, my laptop has a coffee stain. I wish I can double check. Is it? Do you think it's safe not to open it?
Was it working?
It worked, yeah, but they said it's not safe. I don't know what.
How long ago was it?
2 hours ago. Thing is, right now it's very, very sticky
It's gonna be sticky. I have to clean it.
from the inside.
If it doesn't work, then from the inside.
Okay. Okay. Do you have wet wipes?
Yes.
Let's just do a small search because I have this in another file.
بس مثل 44 دي هنا
Sorry.
حتى question ثاني جبتها غلط.
Here we No.
هي والله صدق في روي.
No, let's just look. And how do we think when I have this guy? Shut off.
I don't know.
How do you know what else?
بيتهم يجي سوا؟
And he didn't do it by intention, but he pulled something, the wire fell on the coffee map. That's a good start. It's super sticky there, huh? Okay. Do you happen to have a cloth, like a microfiber cloth? You seem like a tech guys probably have. Guys, keep solving, keep solving. I'm just gonna check this out. Okay, man. So you're right now telling me it should be what?
B.
Answer?
B.
Okay, so let's just check it out. Let's check. I'm trying to ask you for water spray if there's one.
Water spray. I don't know about we have spray, but not water.
Uh-huh. So right now we're Hardy-Weinberg, right, Heidi?
Yes.
Should be unit 6. Can you read me like one word of the, like 2 words of the thing? What's like 2 words of this thingy? Is it
Snapdragons are type of flowering flower.
Snapdragon? Fine, yeah.
As the question?
No, it's not. I just want to find it in the other file. Because I think it's also there. I'm trying. Well, I can't find it anywhere. Look, I wish you're right because
I am.
that would. I wish you because that would make it much easier. But they asked if you look at the answer key, the detailed one.
هي detailed one. العدد الثاني ليش غير شيء؟
Well, maybe they transferred the letter wrong. Maybe. What do you think?
It was empty. So I
Solved it together, it was A.
So he
The
says
last one was B.
the problem is that the college board messed up and had like two answers in a way.
From the college board answer.
What about it?
It says A. But
The other one.
The thing is, in the back it says
Yeah.
like highly confusing. That's why we are confused.
I think in the end they missed a few questions because like the question after it is B, so they probably like.
So
messed
you're
up the
saying,
order.
like the most important part is to know who's right, me or some.
Me.
Because in the detailed answer key, it tells you why it's that one.
It tells you why, right?
Yeah.
Well, it does not for the other one.
which question was it?
39.
Yeah, it's A.
Oh, it's not 39, right? 46.
What?
14.
Are you convinced now or soon or not yet?
It's 45.
Yeah, we're question
No, it's
45.
A.
He's not yet convinced.
Yeah, رقم واحد.
Yeah, but you should check
لا لا
GPT.
تشتري 39.
It's not 39, we're at 45.
No, no, you need the question. We went back to that question.
Oh,
It's A, it's A.
Look, I wish it's B, honestly, man, like it makes my life easier too, but my pride will get hurt a bit, but that's okay. It's not the first time my pride gets hurt with students.
والله مستر كلامك شات جي بي تي منطقي.
Well, I know. No, I'm saying I wish you're right. Like I wish.
Sometimes he's wrong. ChatGPT is not always correct.
This time is correct. Correct.
Why do you guys always call it he?
He, she.
All right. Thank you. Yes. Sorry, not person. Somewhere.
was it Khalil's fault or no?
What do you think, bro?
Yes.
It's fine.
He always wants to like play with the tablet, like write on it. I mean, so he pulled it and he pulled the wire and the coffee dropped on everything, on the floor, on the chair, on the carpet. You know. Okay. So yeah, guys, look, we will do some extra research with that one because the answer key is actually confusing. Because the answer key says A, explains it nicely. Can we just like send the answer of this one to the group, the 39?
The detailed one or the other one?
The detailed, not the detailed, like, yeah, there's exactly that one. Can you send it, just read it so you can see what's going on?
ما عندي الجروب اني.
Sorry?
ما موجود بالجروب.
You're not in the group?
لا.
Can you send it to Justin?
What's his phone number?
That's why you cannot vote. He's not in the group. Okay.
So what's the vote? 2? That's it.
Road is closed. You found it in the end, right? Okay.
You're talking about me?
Did you find the house?
Oh yeah,
Where did you go?
we went to the other road.
Oh,
Yeah, we tried going from the back.
okay. All right, let's continue.
Is the answer?
Sorry?
الدزل ولا لا؟
Yeah, this.
Okay.
Let's continue. So, yeah, do some research at home about this one and let us know. I actually believe it's B, but for some reason they said it's A, and I also explained it well. Can you guys just open your phones for a second? Let's see it. Okay. Did you send the thing?
Yes.
Okay, so guys, if you have your phone open, do you guys have your phones?
I'm on the group. My dad is.
Do you have a number or not yet?
Yes.
If you want, I can add you.
No, I don't have WhatsApp. I have space.
Okay. Wow, says it so well. It's a Hardy-Weinberg equilibrium with only two alleles. Yeah, it's nice. Say. I guess we'll continue. Okay.
346, right?
Yep, which of the following best explains why a cell's plasma membrane is composed of two layers of phospholipids rather than just a single layer?
They're not identical, so C and D cancel.
Anything.
Because look, they appear like this, tail to tail. Okay? 47. A student calculate the average surface area to volume ratio of 4 different cells. Okay, guys, the higher the surface area, the better, correct?
Yes.
What you got?
Question 46.
Yes. Shall I do it?
No, خلاص.
The higher the surface area to volume ratio, the better, which is going to be for the simple squamous. It's better because of more diffusion. More diffusion. The simple squamous. Good.
Easy. It's the ice.
Oh,
By the way, this week again, he made another mistake in the exam. We didn't do it.
he gave you the answer key last time.
Last time answer key. This time.
Who gives the answer key as the exam?
This
Okay.
time,
Oh,
one
the
question
professor.
was only related to the topic given.
Really?
He forgot. He brought something from like 2 chapters ago. He brought the whole exam from 2 chapters ago.
Wow. So you have an exam, you cancel the exam?
This one didn't get canceled. This one, he put like 10 marks on each question that was in the topics included.
Wow. Great teacher.
It's not
Okay.
his fault. It's the school's fault.
Hey, Allah.
No, like he said it in class. It's like this.
Oh.
This is not my fault. It's the school's fault.
Which of the following best explains how the extensive folding of the inner mitochondrial membrane benefits? Guys, if they mention folding, they mean more surface area. Which is where? Which is? Now, someone's going to say, how come it's not C? Is water going in and out of the mitochondria?
No.
I mean, let's say it is. Is that the function of the inner mitochondrial membrane? No. Its function is to make ATP. Okay, energy. Here it shows you a graph where the temperature affects whether the sea turtle is a male or a female, correct? And shows you here that the percent female increases as temperature increases. So female versus male is going to be temperature dependent in a sea turtle. Okay. At temperatures below 23 or above 33, eggs don't become male or female. Good?
What?
At temperatures that are below 23
Okay.
or above 33, eggs don't even develop to a male or a female. Which of the following best describes the results shown in figure 1? What's happening in figure 1? More temperature means what? More percent female. Is that there?
Yeah.
Where? Exactly. Exactly. Which of the following describes a scenario that would result in a phenotypic change shown in the graph? Okay. What type of selection is this? Directional, disruptive, or stabilizing? Stabilizing. Because they're moving toward one big peak in the center. That's called stabilizing selection, where they basically favor the intermediate. So large and small, that's an extreme, non-intermediate, right? So A is off. Okay.
Is it D?
Yes. If they lay a clutch of eggs, which is smaller than average, it's bad. Larger than average, still bad. Because of the high mortality rate. So it is D. Researchers have identified a molecule produced by a marine invertebrate from which the drug trabectedin is produced. Soft tissue tumors treated with this molecule, right, rapidly decreased in size. That's good. In a study, healthy cells and tumor cells sampled from skin cancer patients were collected. Okay. So what's happening here? DNA in a tumor cell is less. Cyclin is going to be low. That's all indication of a messed up cell cycle. Which of the following best explains the most likely method by which this anti-tumor drug works? Yep. Because the DNA goes from 12 to a 6. There's a problem with inner replication. So it's going to be 6. I mean C.
Mister, I solved this one without even reading it. I just read the last part.
Which is?
Just the question,
Really?
and I got C.
How come you didn't say D?
Because it talked about cancer cells, like stopping cells, and this C made more sense.
How about D? Why is D wrong?
It said creating errors. So when I heard creating errors in DNA, I was like, it's wrong because cancer is already creating errors in DNA.
Mm-hmm.
That's how I found out.
It's actually not why. Can someone tell us why D is wrong?
Me personally, I thought it was D.
C.
Guys, it says causing the cyclin level to increase. Well, if you guys look at the table, it actually became less. That's why D is wrong. Okay. We have an inherited disorder that's caused by an insertion mutation in non-coding portion of the FX gene, where the GAA triplet is repeated hundreds of times. FXN gene encodes the protein frataxin. Okay. What type of inheritance is this?
Autosomal.
Autosomal.
Recessive.
Recessive. It's skipping of generations, and gender does not matter. So it's going to be autosomal recessive. A researcher collected DNA from several members of the family and used PCR to amplify the FX gene from each individual's DNA. The researcher then used DNA gel electrophoresis to separate the DNA. Okay. Who knows how to read these bands? If it's a single band, it's homozygous. Okay. A single band is homozygous. So individual triple I4 is homozygous. Individual IV1 is also homozygous. But if it is a double band, heterozygous, double band, heterozygous. Okay, The researcher also used the computer to model the structure of the mutant FX allele. The model suggests that the repeated GAA triplets in the mutant FX gene may lead to the formation of an unusual triplet strand. So a triplet DNA is not normal, right? It's not normal at all. Okay. Based on data and figure, okay, we just said that, right? Autosomal recessive. The probability that individual III5 will develop the disease. III5. Uh-huh. The parents of III5, are they carriers or not?
Yeah.
How do you know they're carriers?
They don't have that disease.
No, how do you know that they're not even carriers? Like, can they be not carriers? Like, can they be homozygous dominant?
No, because one of the parent has the disease.
Not why. It's because one of the kids is sick, right? So because 5's sibling is sick, that means the parents have to be carriers. So now we're gonna have, this is parent 3, and this is parent 4, and they have to be capital A, capital A, small a. They're not sick, but they're carriers. Now, when they do mate, you get 25% small a. You get 50% capital A, small a. 25% capital A, capital A. The chance of being sick is what? 1/4, or simply 25%. Okay. Did I get a question wrong, you said, when I was cleaning the laptop? Or was it just the Hardy-Weinberg one?
I thought 44 شان.
Really? Was wrong?
اي دي كان صحيح.
44. A decrease in the crab?
اعتمدت يلا اسف.
Oh, wait.
Yeah, it was the
Oh, like the question says, increase in the sea slug is caused by what? Is caused by what?
decrease in the crab population.
Yeah, because the crab eats it. Yeah, that's why. So if they were to increase, it's because their predator has decreased. Okay. So it is going to be choice. Where is it? Yeah.
Pure Webster.
No, just one question, man.
2.
It's about that one. We're still debating that one.
Oh.
I swear, I wish you were right. Because I actually had to have like 2 explanations for one thing.
واحد من اذكى طلاب صفنا من قال بي.
Well, I also said B. But then on the explanation, I was like, well, okay. I guess Hardy-Weinberg means 2 alleles. And you know the problem is, there's no other question about this but this one, like Hardy-Weinberg with chi-square. Only this one. That's the problem. They're like, there's no way to check. Okay. Can you ask DeepSeek? Which of the following? It's free. Just install the website. Which of the following statements best describes the results seen in Figure 2? In Figure 2. Okay, so III4 and IV1 are both homozygous, but III2 and 3 are both heterozygous, correct? Yep, heterozygous means what? Carrying 2 different alleles. Okay. Individual IV1 is homozygous, so yeah. And IV1 is sick. He's homozygous recessive. He has 2 copies of the mutant allele. So guys, capital A is not a mutant. Small a is a mutant. Good. '55. Which of the following best describes the most likely effect of the formation of a triplex DNA on the synthesis of the frataxin protein? Can the protein even be made if DNA is not double helix? No. So the protein won't even be made.
Let's see.
So it is C. Exactly. You cannot make the protein, which means decrease in the frataxin mRNA transcription. Which of the following types of bonds is the most likely responsible for the unusual base pairing shown in figure 3? Guys, what bond is between C and T? Hydrogen bond, yeah. Because nitrogen is bases. Okay, I'm just gonna use the bathroom. This will take us like 8 minutes to explain. So if you want, read up in the answer key that I posted. It's detailed. Here's the bathroom or yeah, somewhere.
لا درانك بديلة درانك بديلة درانك بديلة بعد ياخد
تجيب التاني.
Any progress? All right, guys. So, a scientist is studying population of lizards with 3 different color phenotypes. Okay, how many degrees of freedom do we have in here?
1.
1. Hardy-Weinberg is 1, exactly correct. Okay, now we have a capital allele and a small one. Okay. We have 100 lizards. If we have 100 lizards, and each one has 2 alleles, how many alleles do we have in total? 200. Okay, very good. Is that inside the house or? Your neighbors, do they have a car or no? Good, because I parked there.
The 200 part, I didn't get it. What were you talking about?
Allele.
Alleles.
200 is
So, guys, how many capital A allele do we have?
140.
Hmm?
75 times 2, 140.
This is 2, right? Here there are 2, right? So 2 times 75. Here there is 1. 1 times 15. Here there is 0. 0 times 10. Add them all up, you get 165.
Yeah.
So how about the small a allele? We have 200 alleles. So 200 minus 165. Exactly. Let's find the frequency of the capital A allele. 165 divided by 200. 0.825. Frequency of the small a allele, 35 over 200, 0.175. Is confusing?
Yeah. Hello.
Which part are you confused at?
Where did you get 200 from?
We have 100 lizards. Each one has 2 alleles
Oh,
because they're diploid. We did this work somewhat similar to last week's question with the mosquitoes. You were in the trip. Okay. We know the frequency of both alleles. How was the trip, by the way?
Good. I was with Khalid. The stadium.
Okay. Yeah. No more coffee in Summer's house. She barely offers me. This time I accepted. And now. Okay. So let's find. So look, guys, this is P and this is Q. Correct? Let's find P squared. 0.825 squared. Let's find Q squared. So 0.175 squared. Let's find 2PQ. So 2 times P times Q. Let's see how much that is. Zero point 68.
0.825
They're
squared.
both squared though.
No.
It's okay, man. We all lose one day. It's okay to get humbled. 0.29.
That's
Now, can we just add them up and see if they equalize to a 1? Stop trying, man. It's okay. I've been there before. Okay, just today, actually. How much do you get if you add them?
1.9.
Okay,
One.
humbled again. 1, bro. What the heck? If you get a 1, that's okay. That's actually correct to get a 1. You must get a 1, right? Okay. Why? Why should you get a 1? Because P squared plus 2PQ plus Q squared has to be a 1. Now, we have how many lizards again? 100. So this times 100, this times 100, this times 100.
3%, 68%, and 29.
3%, you mean 68 lizards, 3 lizards, 29 lizards. So now come here. Sorry. Is it outside or inside?
Outside.
The fuck's it? What? Outside? It's coming from inside. Okay.
Going to math.
Grade 2 math.
Yeah.
Number. So. 68 for the P squared.
Recording, right?
Yeah. Shall be published to the world. Because, you know, a lot of people watch these recordings. A lot of people.
I think no one does.
I actually do sometimes. Like I watch and like I cut parts, you know, like make sure it's perfect. So whoever decides to watch it one day.
What?
Now look, let's find the chi-square. This is all for one question, guys. All for one question.
أنا أفضل الامتحان.
I
think
swear.
guessing is the right thing to
And
do.
this, I swear,
4.9.
it's actually what?
Actually,
0.9?
no, I just guessed 1.9.
Okay. Observed minus expected over expected. Observed minus expected over expected. Observed minus expected over expected. You get 23.8. Not even the exact answer any after all this work. Okay, let's go back to the question. 52. I mean, 58. Scientists compared the chemical structure of several molecules that various bacterial species use for quorum sensing. What's quorum sensing? Ability of the bacteria to detect the number of related cells nearby. We have three chemical structures here. Which of the following will best guide the investigation for the link between the structure of single molecules and the evolution of quorum sensing? Guys, the best way to know if two species are related is to take one substance from one of them and put it in the other one and see the same exact response. Okay? So, which one do you guys think it is?
Me.
Do the species evolve from a common ancestor? Investigation between the link. Okay. Yeah, it looks like because you have a common ancestor. Okay.
Or
Between
similar
us.
signaling molecule.
Similar
Signaling molecule.
Yeah. The structure of the signaling molecule and the evolution of quorum sensing. Did they evolve from a common ancestor that uses a similar signaling molecule? Yeah. How about C? Why is C wrong?
Because a
Concentration
variety of
علاقة.
different molecules can evolve to perform a similar function.
Like saying high enough threw me off, right?
وحتى شو يسمونه ماكو دليل على evolution.
Sorry? Yeah. They're not saying evolution.
Yeah.
They're not saying that. Okay. The best one is going to be choice B. What did the answer keys say, like exactly for that one?
Which one?
For B, like why is B right? Like the exact answer.
It says B as
ما شو ذا
Oh. Bacteria species likely have similarly structured signaling molecules as a result of common ancestry. That's right. Similar structure because of what? It's a homologous structure, right? Very good. Okay. Yeah, because we have three different molecules. Okay. All right, good. So it's going to be choice B. Carbon dioxide most likely enters a cell through which of the following processes?
simple diffusion through the membrane.
It's nonpolar, right? So simple diffusion. In pea seeds, yellow color is dominant to green color. Round shape is dominant to wrinkled shape. Okay. We're mating this with this. You get this guy and this guy. That's the F1 generation. Okay. And if you cross them with each other, you guys know what you get, right? 9, 3, 3, 1. Okay. Good. So the 9 is going to be the yellow round. 1 is going to be the green wrinkled. Which of the following best explains the results? So we got the right results, correct? There is no linkage here. Right?
They are sorted independently.
Exactly correct.